Yeah, basically, this is the article that I'm going to share today. And Ambas analyzed for SASP in different conditions on genotoxic stress. And basically, it just identified what components it contains in different conditions of cell senescence. And then mainly these articles discover common components in different cell senescence phenotypes, and they identify protein that exclusively exists in those phenotypes and define 3 key signaling pathways for causing different kind of to cause cell senescence by different way. And then so basically how they do it, firstly they have 2 types of cell line, bone marrow and adipose mesenchymal stromal cell. And then they have 5 different cell senescence conditions. One is irradiation in high dose and low dose, and doxorubicin and hydrogen peroxide and replicative cell senescence. The first 4 are an acute cell senescence condition, which they do the treatment directly. And the last one is basically having 30 days cell culture on the cell and causing chronic cell senescence. And they treat the cells and create 10 different cell senescence condition and do the experiment. And then basically when they finish it, they firstly use galactose assay to calculate the percentage of cell senescence within those conditions. And then they use a mass spec to identify what components it contains in the protein secretome. And then they use gene ontology to identify the molecular function, the biological process and cellular components within different pathways. And then finally they use a analyze called IPA to identify the common pathways that occur in different phenotypes. And I'll break it down one by one. And so basically the first one is the beta galactose assay, and then top left is we can see including control, it got 12 conditions. And this is the picture under the microscope, and you can see control got more like cell containing because they didn't undergo cell senescence, but the remaining one basically they all got cells, they induce cell senescence. And the blue staining one is basically the maltubes, and the bottom left is the mean of the cell senescence is different condition, and the right one is the total number of proteins that define it after they induce cell senescence treatment. And you can see adipose MSC, that's a ribosomes, contain the most protein after the treatment. And this is the common pathway that they classify a lot. They classify by using IPA analyze. In total, there have 138 of canonical pathways, and they classify as they mainly break it down for actually it's 4 groups. The left one is an extra one, but is miscalcaneus basically is classified as other categories, but they mainly classified as metabolism and extracellular matrix or cytoskeleton class, or also redox pathways or gene expressions. And they can identify the overlapping networks by using IPA, basically just see the common network that occur in those 10 types of cell senescence. And the things that I highlight is basically the overlapping networks that occur in 9 out of 10 cell senescence conditions. And the main point for this page, I would like to focus on the gene expression one, because ubiquitin pathway is contained or basically exists under all types of cell senescence pathways, which is quite interesting. And then by using the GeoAnalyze, we can understand the biological process and molecular functions. This one we can just like go through a bit, but not very details. But yeah, as the previous IPA-Analyze show, I think you can mainly focus on regulator of gene expression, because basically we know ubiquitin can modify the DNA changes by translocation and do very many types of things. And I just thinking, can we use any like type of dubs? Or I have no idea yet, but I just think about can ubiquitin contribute, or having some like I can discover some unknown signal pathway or signaling, and by reading these articles and do some experiment. And yeah, and then this is two I think it's quite complicated paragraph for me, but I just break it down. And let's go to the left one. The table 2 is basically show the common networks that exist in different types of MSC. In adipose MSC, if you go read from strict, AMSC is the protein that occur here is the protein that commonly exists in all types of AMSC in those 5 cell senescence condition. And yeah, bone marrow one, MSC, and the common one, they basically just overlap, and we can focus it on the right. The common one are the overlapping networks for different kind of cell senescence condition. And yeah, the right one is the 11 protein that exclusively exists in all types of cell senescence phenotypes. And then it's I just bring it out as these are the main results within the articles. And yeah, I think this one is relatively important. And then these 9 proteins is belongs to those 11 proteins that I just mentioned, and they induce cell senescence in 3 different kind of signaling pathways. And the first one basically is just remodeling the extracellular matrix by using MMP2 and TIMP2. What they do is basically MMP2 is just promote and regulate inflammation process, which causing cells to grow. Meanwhile, TIMP2 it just negatively regulate the cellular response and growth factor, which does the opposite thing with MMP2, and they have basically one positive and negative regulator in remodeling the ECM, and then it just basically causes cell senescence. The second one is the pyrosine senescence signaling, and yeah, PAI1, and yeah, insulin binding growth factor, yeah, protein 3, or yeah, type it wrong, it should be P3, and basically just causing cells, the senescence cell to implode into like all the cell and causing its neighbor cell to become senescence and other types of senescence. And the final one is basically these are the proteins that exist in gene expression 1, and yeah, it just basically this protein just making the cells become more resistant toward apoptosis, as senescence cell will not be apoptosed. Basically you're just making the cells survive under stress and become apoptosis resistance. And then yeah, I taught a lot, and yeah, I just want to conclude that for these articles basically find all the proteins that produce under cell senescence in 10 different types of phenotypes. And then they classify 5 classes of SASP components, which including the miscellaneous one. And then, yeah, by using GeoAnalyze, we can know the biological process and molecular function of each group on those 5 classes. And they also identified and showed the overlapping networks and those 11 exclusive proteins that they found in the cell, the senescent cell. And yeah, they point out that those proteins can involve in 3 different pathways for inducing cell senescence. And then, yeah, I have no idea about how this article can really contribute to my experiment yet, but I just got some inspiration after I read it, because yeah, ubiquitin pathway is the only gene expression pathway that exists in all types of cell senescence conditions. And then the article also point out that those exclusive proteins and overlapping network, that it just exists in ubiquitin pathway. And they point out a lot of proteins, but meanwhile they haven't really understand all the proteins and how they haven't understand how those proteins evolve in those involving the pathways. And yeah, maybe I can use them as marker, or I don't know. I don't have any idea yet. So I know I talk quite fast, and yeah, this is basically the end of my sharing. So yeah, thanks guys.